And we are live. Hello, everyone. Good morning, good afternoon, good evening, wherever you are. Welcome to this new edition of the Worldwide Neuro Forum, and the latest one, special Christmas edition, as you can tell already before I've introduced today's speaker, Matthew Cobb. Thank you for being here. It's really a pleasure, and the timing is just perfect for us. I hope it's the same for you.
No, it's fine. Thank you for the invitation. It's great to be here. Hello, everybody.
So, little words about you, Matthew, before I let you on the front of the scene. You did your PhD at the University of Sheffield in psychology and genetics, looking at the mating behavior of 7 species of fruit fly. I was impressed by the precision of that specific number.
Very important. Very important. They're all closely related.
First postdoc at the Institute of Psychiatry in London, where I quote, "I spent my time getting twins drunk," unquote. This was interesting, but convinced me that I did not want to do research on human beings. I'll let you add to this if you feel like it at some point. After that, Matthew went to the south of Paris at the Institute in Gif-sur-Yvette, where he got acquainted with and expertise in chemical communication, how animals communicate with each other, in particular through smell and pheromones. Set up his own lab in the French system using Drosophila maggots as a model organism, and also studying chemical communication in ants. Went back to the UK in 2002, where he took up his position as a lecturer at the University of Manchester. In 2006, he published his, I guess, first book, titled The Egg and Sperm Race, 17th Century Scientists Who Unraveled the Secrets of Sex, Life, and Growth. Several books, successful books since then, successful in terms of attracting interest from the scientific and broader community, but also in attracting and receiving awards. Other books relating to life sciences, Life's Great Secret, there's the race to crack the genetic code, and also some books of non-scientific history relating to the French resistance in particular. So Matthew just finished writing a book about the history of the brain. It's a great pleasure to be able to welcome him on this occasion. The book is called The Idea of the Brain. So it's a book, as the title says, about our ideas about brain and behavior. It's not a linear narrative of the sequential discoveries that led to our understanding of the brain, but rather a much more dynamic account of a topic-by-topic investigation of approaches and emerging ideas of how the brain works. I really love the book. I told it to Matthew, if you haven't read it, seriously consider doing it. There's usually some time for reading around Christmas. It's anecdote-rich. It's extremely seriously documented. I really think it's what you would get if Malcolm Gladwell and Jared Diamond sat together to write a book. And I really put it to that level.
High praise.
Yeah. It's really fun, and there's fun facts, and there's real facts, and it's well documented. Every student out there, in my opinion, should read it. It's a great way to know where we're coming from as neuroscientists, and get some insights even on where we might be going. So with this, I'll stop talking. Matt, Matthew, a really pleasure to have you, and the stage is yours.
Okay, so I click on share screen.
Yes.
Allow, and then I'm going to play in here. Oops.
Yes, and then full screen.
There we go.
That's
perfect. That good? Yep. Okay.
All yours.
Thank you very much, and hello everybody, wherever you are around the world. I'm going to be explaining some of the key ideas in my book and some of the key people. And there'll be some advantages, actually, for those of you who already seen the book, in that read the book or listened to it. There'll actually be some videos as well, which, of course, we don't have yet. So I think the starting point is the truth that, the brain is a bit odd, because for most of our history, most people have believed that thinking is done with the heart, and the brain wasn't seen as being anything very important. And we can see this in languages. Just look at all these phrases from English, and similar phrases exist in every language. These are all everyday phrases that you would use, but it's the heart that's the focus of them, not the brain. If you put brain into any of those phrases, it just sounds utterly weird. And so in a way, they're not simply kind of linguistic fossils. They're actually telling us something true, that the reality is, as a paper from the 1990s had it, the brain has a body. Our brain is not just sitting up there in our heads. It's connected with our visceral activities, and everything we feel is associated with the way that the body's moving and interacting with the brain. And that's why these phrases still exist, because they have that intuitive power that goes beyond whatever we might know about things happening in the brain rather than in the heart. So how do we get to this point? How do we understand this? Well, as often is the case, for the first kind of systematic recorded dispute about this, you need to go back to the ancient Greeks. And Aristotle, brilliant thinker, but he was absolutely convinced that the heart was the center of thought and the brain was simply something for kind of cooling stuff. It didn't seem to have didn't move. The heart moves. One of the main ways that the Greeks thought about the world was things with motion were very important. The heart clearly is doing a lot. The brain just seems to kind of sit there and have no power of motion at all. So he argued that it was acting to cool the blood, and the blood was the key thing that was moving what they call the animal spirits about. And some of you may have come across that term, animal spirits. It's got nothing to do with animals. The animal bit comes from animated. It's the same root. It's to do with movement and creating movement. But then during the 4th to 3rd century BC, so after Aristotle, a number of thinkers began to challenge his ideas. Herophilus, Erasistratus, and Hippocrates, or the writers who wrote under the name of Hippocrates, they all argued that the mind was in the brain, and the brain was the bit that doing the thinking, not the heart. But none of them had any actual evidence for this. This was all based on arguing about it. Hippocrates, for example, claimed that epilepsy, which was called the sacred disease for reasons that remain obscure, that this was a disease of the brain. But whoever was writing under the name of Hippocrates had no proof of that. The key proof began to be assembled by Galen, who wasn't an ancient Greek, but an ancient Roman. So he's living in the early years of the Common Era. He's a Greek citizen, but he was born in what is modern day Turkey, and he was part of the Roman Empire. So it's all kind of complicated. And he did something rather gruesome. He did an experiment, and he did experiment, I'm afraid, on a pig. And you've got to remember, this is before the time at which they had anesthetics. So he got a pig, and he wanted to disprove the role of the heart. So he had a disputation. And he insisted that his opponent, they opened the poor old pig up, and despite it being chained down, it's clearly making a lot of noise. And he made his opponent put his hands around the pig's heart and stop it from beating. And of course, the pig carried on making a noise, despite the fact that its heart wasn't able to move. However, if you opened the head and pressed on the brain, then the pig went instantly unconscious. So Galen, by this simple experiment, very unpleasant but simple, had demonstrated that the heart and the brain had very different roles in bodily function, and that certainly in terms of controlling the voice, which was seen as being one of the main features that the brain could do, it could control the voice, the brain was the thing, not the heart. Now, although this idea was well known and this experiment was well known, and Galen's writings were incredibly influential, and he's at the origin of what passes for medicine in the West for, well, about 1800 years. So he's an amazingly influential person. Nonetheless, because of the impact and the power of those everyday sentiments that, in fact, your heart is moving when you get excited, not your brain, most people, even thinkers, didn't accept Galen's experiment as being decisive. And Aristotle's followers were entirely unconvinced, and Aristotle was carried on being significant in Western thought for centuries and centuries. And we can see that this uncertainty about what was going on in Shakespeare. Now, Shakespeare's plays are often seen as being quite kind of fancy and complicated these days and only for intellectuals. But of course, at the time, they were watched by, they were kind of the they were the telenovelas of the day. They were watched by the ordinary people. And in Merchant of Venice, which was written at the end of the 16th century, he has one of the songs has this little opening: "Tell me where is fancy bred, or in the heart or in the head." So Shakespeare is making one of his players sing this song, which is highlighting this intellectual uncertainty that was going on at the time. And it would have been recognized by the groundlings. They were the people standing up in the Globe Theatre in London, who would have been, they were talking to each other, throwing vegetables at the stage, all sorts of stuff. And they would have got this joke. They would have understood that these clever thinkers, they still don't know where fancy is. Is it in the heart or in the head? We don't know. By the 18th century, the role of the brain had become accepted. There was no brain-centric moment. There was no decisive experiment, but accumulation of evidence led this man, Priestley. Priestley was an astonishing chemist, thinker. He did loads of experiments about what we would call neuroscience, trying to work out how neurons worked. And he said, in my opinion, if you can do English accents, you've got to imagine him having a very strong Yorkshire accent. I can't do it, so I can't do it. But the Yorkshiremen have the reputation in the United Kingdom of being very opinionated. And he said, in my opinion, there's just the same reason to conclude that the brain thinks as that it is white and soft. In other words, it's just obvious. There's no instance of any man retaining the faculty of thinking when his brain was destroyed, and whenever that faculty is impeded or injured, there's sufficient reason to believe that the brain is disordered in proportion. So for Priestley, this was just kind of obvious. The heart's a pump, the brain is doing the complicated stuff. But as I said, there was no single moment. There was no decisive experiment. It was just this slow accumulation of certainty from the 15th century with the first really detailed dissections, which showed that the brain was complicated and the heart was just a pump, right up to the 18th century when, at least for thinkers, this became obvious. For the general public, the ordinary person, and in particular for the world outside of the educated West, most people carried on thinking that it was the heart or the liver or some other part of their anatomy that was the center of thought. And really the theme of the book and the theme of what I'm going to go through in this little talk is how thinkers at various points used metaphors from technology to think about what the brain does. And the first person to do this was Descartes. And in the 1630s, he did a lot of dissections. He wasn't just a thinker. He did dissections of the brain, worked out its organization and so on. And he was very struck by these moving statues, these hydraulic statues. It's kind of crap animatronics, where dragons would flap their wings and move as weights or water pumped up and down. And this character here would hit the dragon on the head with a club. And Descartes thought, well, maybe that's how nervous systems work. Maybe that's what's going on. And he has this very famous model whereby if you burn yourself, fire helpfully marked A, you'll burn yourself on your foot marked B, and then the animal spirits would move up in a pressure up to the brain marked F, and then down would come the response because it was reflected. That's where reflex comes from. This goes up and is reflected back, and you move your foot away. Now, Descartes' hydraulic theory was pretty smart, but it was fairly easy to demonstrate that it was wrong. And as soon as his book was published after his death, because he was rather embarrassed about it and worried about the church and so on, so it wasn't published in his lifetime. Once it's published in 1660, immediately anatomists start getting frogs' legs and dissecting out the nerves and chopping the nerves. And you know what? There's no kind of hydraulic spurt as there would be if you were to cut one of these pipes feeding the dragon. So whatever was going on with nerves, it wasn't the kind of pressure-based view that Descartes had. But, I mean, he deserves enormous credit for looking at these statues and thinking in that way, because similar statues have been around in antiquity, but nobody had actually put the two things together, thought this could be a model for behavior. By the end of the 18th century, there were really quite elaborate and extraordinary mechanical devices had been created. So, for example, this is the writer. It's this little boy here. He's got 6,000 parts in him, created by the Swiss watchmaker. And this is still on show in Neuchâtel in Switzerland. So after this is all over, go to Neuchâtel and you can see this in the museum. We're going to see a little video of this thing moving. So you can program it. You can put different letters in and make it write different letters to form words, to form a letter. Look at those eyes flicking, and that eerie. Now, nobody thought that this was alive. Nobody thought that we have cogwheels in our head. But the idea that there might be possible to create machines that would imitate and even represent in some ways the same way that the brain was working began to grow. However, the big change came with the discovery of a mastery of electricity in the 18th century. People were able to show that you could get movement in frogs. And then from the beginning of the 19th century, people, in fact, modeling, trying to work out how animals produce electricity. Volta produces what he called his pile, what in English we would say battery, but you still say pile in French, for example. And this was the battery was based upon the model of how the electric eel produced its electric shock. And Volta interlayered various bits of metal and acid, and he was able to produce a continuous current rather than the sudden discharges, which is all people have been able to do in the 18th century. And as a result of that, you could then make movements take place. And this is a very famous event that took place at the beginning of the 19th century, where an English criminal, he killed and he murdered his wife and his child. Immediately after being hanged, he was brought into a room, and then these batteries were applied to either side, electrodes applied, to see the batteries down here, applied to either side of his head. And of course, his eyes opened, muscles started to move, his arms moved, and people were absolutely terrified. And it's very striking that a similar kind of demonstration using animals was shown at the Royal Institution by Humphrey Davy in about 1810, 1812. And a young teenager called Mary Godwin was apparently there. And it is thought that this demonstration of the apparent power of electricity to recreate life was at the basis of her book Frankenstein, which she wrote under her married name Mary Shelley. Must be said, there's no mention of electricity anywhere in the book. But anyway, this is the kind of thing that it was apparent that electricity could produce movement, and even perhaps was the basis of life itself. And here's an example of some of the experiments that were done by Aldini. You could actually wire up two cows' heads together, form an arc from the ear of one to the left ear of the other, and saw with astonishment that two heads make horrid grimaces, so that spectators who had no suspicion of such a result were actually frightened. Well, you would be. I mean, it's quite terrifying, the kind of thing you could do. But this is still quite uncoordinated behavior, and you hadn't been able to recreate life. By the end of the 1830s, electricity had now been mastered and used for communication, and you began to get telegraph systems covering the whole country. This is the United Kingdom in 1846, and you can see that everything, of course, infuriatingly for somebody who's based in Manchester, which is up here, everything goes down to London. That seems to be the head. And parallels were drawn between the telegraph system and the nervous system, such that both people thought, well, maybe the nervous system works a bit like the telegraph system, and maybe the telegraph system, with its sending out of news and getting of news from the periphery to the center, is working like a body. So that this was literally the body politic in 19th century Britain. And the man who actually developed this most was somebody I'd never heard of until I started working on the book, Alfred Smee. And he's kind of British scientific aristocracy. One of his descendants currently works on Nature. I think she's an editor there. And another one is an academic at Cambridge. So he's left a remarkable influence. But what he argued in a series of rather odd books is that we really have electro-telegraphic communication in the nervous system. That which is seen or felt or heard is telegraphed to the brain. And he produces these fascinating kind of really, tantalizing pictures about how nervous systems work. So here we've got a lower animal. You've got muscles, you've got skin, ears, eyes, you've got body, nerves, and brain. And you've got what looks a bit like the kind of models that modern neuroscientists use to go into the brain. Now, what Smee argues is that this shows, apparently, how the idea of a nest may be implanted in the bird, or of a honeycomb in a wasp or a bee. Now, I can't quite see it myself, but he insisted it was the case. And humans, of course, just have a bit more kind of connectivity. What's striking is that although he thought that this was all about electricity, the model that he used was essentially hydraulic, and there's no electronic component. I mean, that's because the technology hadn't been developed. It was still extremely crude. He knew that there was something electrical going on, but how the organization could produce behavior, he didn't actually say. And above all, what's very striking, there's no localization of function there. Yeah, this is all kind of abstract. Apart from the fact you've got ears here, then it all gets connected. But at the same time, in popular culture and for some parts of science, what's called phrenology, the idea that you could show how human behavior could emerge on the basis of feeling the outside of somebody's skull. So the idea was that there were lumps on your skull, which there are, and that these reflected organs in the brain, which of course it doesn't, because the skull's really thick. But nonetheless, everybody in the 19th century believed in this guff. Queen Victoria to Karl Marx. So it was the whole of the political spectrum. Every British, European or American intellectual believed in this. Sherlock Holmes, when he first encounters Moriarty says something very rude to him about the shape of his head, because Moriarty, in the books of Arthur Conan Doyle, believes in phrenology, because everybody did. And some justification for this appeared to be found by David Ferrier. What David Ferrier was able to do was to prove that there was localization of function in the brain, not according to the rather crazy ideas of phrenologists, but by using very, very slender electrodes and very delicate electrical currents, using batteries developed by Smee, actually. He was able to show that these different areas in the brain produced responses in the body of a living monkey. And by comparative anatomy, he said, well, this is the same kind of structures in a human brain. He wasn't able to do experiments in the human brain, although in the book I do detail one horrific example of an experiment that was done on a poor patient. But of course, there's one area that's missing between here and here, between a human and a chimp, and that's what I'm using now, and that's Broca's area, which is here, which controls speech and is unique to humans. There's no equivalent to it. And Broca was able to discover this to his great surprise, because he was a good Frenchman and he thought, like Descartes argued, that the brain and the mind were both singular. To his great surprise, in the 1860s, he discovers that there's a link between stroke, the location of a stroke, and damage to speech. And that if on post-mortem, you could discover there was damage to this area of the brain, you could be certain that the patient had been unable to speak. So the localization of speech became absolutely certain. And this is still, to be honest, one of the few areas that we can be really, really sure that there's localization, although how that works, we have absolutely no idea. And around in the final decades of the 19th century, people started to think of the brain as being much more active. Helmholtz, one of my scientific heroes, most extraordinary man, he works out that the brain is making inductive conclusions about the world, that we are interpreting the world. It's not just obvious, visual images don't just pour into the brain and are then seen. You haven't got a little homunculus looking out at you, but instead the brain is active. It's working out what's there. And he figures this out because he notices that we've got a blind spot. And you can demonstrate the blind spot by doing simple perceptual experiments. But of course, we don't perceive it in everyday life. In everyday life, our brain just kind of blurs out, says, well, there's something over there. We won't pay any attention to that. It makes the induction that there must be something there, even though there's no signal, because in that part of the retina, there are no cells detecting light. It's where the optic nerve goes through the retina. You've also got Morgan, who starts to use terms like control, that the brain is controlling things. And the primary aim, object and purpose of consciousness, Morgan argues, is to control what's going on. So the brain stopped being this just repository of sensory information. And now it's actually actively doing things, but in ways which neither Helmholtz nor Morgan could begin to imagine. By 1880, there was a new technology which surpassed the telegraph, and that was the telephone exchange. And when I talk to students about this, they have no idea what I'm talking about. Maybe some of you don't either. So just to briefly explain how a telephone exchange would work, that you would pick up the phone and you would talk to an operator who was normally a woman, and a light would come on here. So she would connect this cable to your slot so that you could talk to her. And you would then say, I'd like to speak to Whitehall 1213. And she would then connect your cable to the slot for Whitehall 1213. If you wanted to talk to somewhere else, to somewhere in Lincolnshire, then you would have to be sent off to another. She'd connect you to a different board that would take you off to a different exchange. So the key thing about the telephone exchange is that it's flexible, that it's not hardwired. You can move around. And so it's got the capacity, at least in terms of a metaphor, for learning, for changing, for altering your output depending upon the input. And this is obviously much more about what people thought about what the brain might be doing. However, people still didn't quite get this. So in 1916, Arthur Keith, who in fact was he was an anthropologist. He was an absolutely terrible racist, and he was also one of the people involved in the false Piltdown Man scam, if you know about that. Anyway, in 1916 he gave one of the key British moments of public engagement with science, which went back, in fact, to the events that the lectures that Mary Shelley saw in the early 19th century. It's at the Royal Institution, and these are the Christmas lectures. And what happens is a big cheese gives talks for children. It's still going on. This year, it's all about geology and climate change and so on. And Keith wrote his talks were all about the machine, the brain as the body as a machine. So he gave each lecture was about a different aspect of the body. And he argued that the brain was like a telephone exchange. And he actually has these little pictures of a telephone exchange here. So you can talk and you can go through and you talk at the transmitter, it goes up, and then you talk in the receiver. And he said this is just like a reflex arc where you've got a skin here, and if you've got a scratch receptor there, then you go around here, and if you're a dog, you will then start scratching. But of course, that is how a reflex works, but it's not how a telephone exchange works. He actually missed out the key point about the telephone exchange, which is it's flexible, and you can connect this connection here with this one here to talk to somebody else. But you can't suddenly start connecting different parts of your nervous reflex. That's the brain that does that, not the spinal cord. So there's no flexibility, and he doesn't even address the question, well, what's actually going along these nerves? Now, around about before the First World War, people got very interested in feedback. And the word hadn't been invented yet, because it only comes into language after the First World War with the development of electronics. But this electric dog was built by Meissner. And in fact, the dog called Celino, rather than being simply some kind of funny device, was that they were trying to develop homing torpedoes. So there's a very serious aim behind this. And how Celino worked, he's got two photoelectric cells. You can probably just see there's one here and then another one here behind this divider. So there's a divider. So if you shine a light, you can only activate one of the photoelectric cells. And he will then move towards the photoelectric cell. Okay, so it's very simple positive feedback loop. He's attracted to the light and will move the wheels. There's a little wheel at the back which should change it. So the electric dog will kind of chase you if you shine the light on it. But I want to show you something even more amazing than that. And this is from Lotka. Now, some of you may have encountered Lotka in courses on theoretical ecology. Along with Volterra, he's the person who came up with the cycling of predators and prey. But he wrote a whole textbook in the 1920s about everything to do with biology. And he was very, very struck by this little ladybird. In fact, I've got one here, made in Slovakia. I'm not going to make it work because it carries on making a terrible racket. The key point is that this is clockwork. There's nothing in here but clockwork. And I'll show you a little video of what it does. So it avoids edges. Whenever it comes to an edge, it will turn round, carry on going, then it meets another edge and it will turn round again. But I repeat, there's nothing electronic in here. There are no sensors. There's nothing except one motor driving it forward. So how does it work? Well, it works like this. You've got driving wheels here, and then you've got the antennae here that keep this wheel, and we're looking at it head on. This is a free wheel. It doesn't have any motor power to it. It keeps that off the ground. So the animal will just move forward. Yeah. But as soon as the antennae fall off the edge, because it's coming to an edge, now this wheel hits the ground and the forward motion is now converted into a sideways motion. Yeah. And it turns in that sharp angle that we saw. And Lotka says, what's going on here? And this is quite remarkable, I think. He says that the toy, the whole thing, is construing the information that is coming from here. There's no direct signal, there's no electronics, there's no nothing, but the whole thing works. You get an emergent property out of three very, very simple components. In fact, only really two components, that one active and then the conditional. Is this on the ground or not? And if it's on the ground, it goes in a circle. If it's not on the ground, the animal moves straight forward. And that's absolutely astonishing. Around about the same time, this man, Edgar Adrian, those of you who are paying attention in the early pages of your textbooks might remember his name, or if you're an electrophysiologist, you'll definitely know his name. Edgar Adrian, one of the most remarkable people I bumped into, not only won the Nobel Prize, his students won the Nobel Prize, his son went on to become an FRS, he became Vice Chancellor of the University of Cambridge. But what I want to focus attention on is his work that was involved in publicizing, in doing public communication, communicating his ideas. So he makes some really important discoveries about how neurons function in the 1920s using amplifiers that were developed during the First World War. And he was able to amplify the signal from a single neuron. He was able to power down the nerve in the stretch nerve in a frog muscle, and then to, as you can see here, record from it and put extra weights on it. And what he's showing is that although the actual signal, the spike, is either there or not, the response of the neuron is not digital at all. It's analog. The more weight you put on it, the more spikes you get. So there's an analog component. And in his books, writing for the general public, he starts to use these words, which still didn't have precise mathematical meaning, but which were useful metaphors to explain what was going on. He talked about information, messages going down the neurons, and codes. In fact, there being a neural code, which we can see here. He's the first person that I've seen who uses these terms, and it's very striking. He doesn't use them in his articles. He uses them for the general public as a way of simplifying and expressing what he's discovered. He even did it on himself. And here he is. He was able to record. So people started to record EEG recordings. So he would do this. He'd do this at conferences. Rather than show a slide, he'd sit down, put the electrodes on his head, and he could show the alpha waves, the Berger waves, as they were called after their discoverer. So if you shut your eyes, you get these fantastic alpha waves. If you open them, they disappear. You shut them, they come back. And he could actually, this is who EDA is, Edgar Adrian, it's him. And he even showed you could do this with a water beetle. So he's got a water beetle, which is a beetle that lives in the water, if you don't know. And he puts electrodes into its head and he puts it in the dark and you get lovely alpha waves. You turn the light on, it disappears. Put it in the dark, goes away. You could even get the effect in humans if you said, okay, keep your eyes open and now do some mental arithmetic. Then the alpha waves went away. I don't know what happened with the beetle. I suspect that didn't work. You had to turn the light on. By the 1940s, people, this person, Kenneth tragically died in 1945, an accident in Cambridge. He was knocked off his bike. But he wrote a marvelous book, very short book, in which he's thinking about what actually nervous systems and the brain in particular does. And what he says is the fundamental feature of neural machinery is its power to parallel or model external events. In other words, it's not just like Helmholtz argued that the brain is in some way intuiting things, it's working things out about the brain, about the world. In fact, it's got a model in there. And by working out what might happen if I act upon the world, you can decide what to do. And I think Craig's insights were absolutely extraordinary, changed the way we thought about what the brain does. And the other two people who changed about not only about the brain, but they changed our whole world are McCulloch and Pitts. Some of you will know of them. Pitts is the most extraordinary person. Somebody said there is no biography of him, and all accounts of his life defy belief, because he never had any degree of any kind, yet he's one of the most brilliant mathematicians of the middle of the 20th century. And McCulloch and Pitts, in a very significant paper in 1943, they said, well, you know what? The way that synapses are wired up, there's logic there, because we could have an ordinary synapse where neuron 1 fires and that produces a response in neuron 2. Okay. But we might have them wired up such that this neuron 3 only fires if both 1 and 2 fire. Or you might have it wired up so that either it will fire only, or either, I'm getting mixed up, either if either 1 or 2 fire. Or you might have it wired up such that 1 fires but not 2, then 3 will respond. In other words, they argued that the way that nervous systems are wired up is logical, and there's an immanent logic in the nervous system. Now, those of you who work on nervous systems will be going, that's not true, and it's not. But they thought it was, and that's all that counted, right? They thought it was. It's all that counted because John von Neumann, the man who developed the devices we all use today, the digital computer, not the analog, imaginary thought machines that Turing was thinking about in the 30s or came to Manchester and started working on in the 1940s. The digital computer was devised by von Neumann. And von Neumann explicitly says in his pitch to the American government to give him loads of money to build this device, he says, I'm going to build a device, a computer that is like a brain, because it's going to work along the basis that McCulloch and Pitts say the nervous system works. So we now say the brain works like a computer, but at the beginning, the computer was a brain, literally. That's what von Neumann thought he was building. Now, he very soon realized, one, that nervous systems aren't wired up like McCulloch and Pitts claim. Secondly, that if he was going to model the brain in a computer, it would be, with the technology of the time, it would be bigger than the known universe. So he kind of backtracked from that idea. But I think that's a fascinating twist on how we think, the role of ideas and metaphor in science and computing went. At the beginning, the computer was a brain. So this is basically where we are. I argue that since the 1950s, we've been not stuck, we've been using the same ideas about what a brain does. It contains symbolic representations of the outside world, and I would argue the inner world, that it manipulates through computations of some kind, which are very hard to define. Those computations enable the brain to predict what will happen and to produce appropriate behaviors, to make the body do things. And among the processes involved are feedback, inhibition, probability calculations. That's basically, I think everybody can agree, this is what the brain is doing. But how does that actually work? How are these processes represented in our heads? Because neurons aren't digital. They've got that analog component. Anybody who's worked on nervous systems knows they're analog, sensory neurons in particular. And synapses are just mind-bogglingly complicated. They're not those single on-off things that poor old McCulloch and Pitts claimed. There are dozens of neurotransmitters. There's activation and inhibition. In the human synapse, there are 5,500 different proteins. I mean, it is just mind-bogglingly complicated. You have hormonal effects that over the long term or medium term can alter, potentiate whether or not a synapse is going to fire or not. So this is not at all like a digital device. It's not like what we're using now, computers. These are completely different. And I just have to show, this was from in EMBO Journal. So this is live imaging of just 45 proteins. Remember, there are 5,500 in your synapses now. Look at the time scale as well. This is in milliseconds. And so this just this complete chaos, this mad chaos going on in our synapses all the time, which is just unimaginable if you try and model that or think about a computer metaphor. It's just completely inappropriate. Here's a single maggot stretch cell. So all that maggots do is move forward. I've spent a lot of time looking at maggots, which is why I'm interested in. So they need to know I've stretched. They've got a little signal in the neuromuscular junction. This is not even its brain. Neuromuscular junction, which says, oh, I've stretched. We can get ready. Let's do it next. Okay. And this is what the cell looks like with GFP in it. So each of these stretch receptors has 53 input synapses, 18 output synapses, and is connected to 74 other cells. Many of those synapses are polyadic, so they've got more than one neurotransmitter. And that's just for a maggot in one of its segments, repeated over and over again, to say, oh, I've stretched. That, as far as we know, is all it's doing. That is the kind of amazing complexity you have, even in the peripheral nervous system, never mind in the brain, which is down here. And this is tweeted by Sophie Scott, who works on hearing. And she was getting to grips with how hearing works. This is the human hearing. Spent today reading about subcortical auditory processing, like this bastard, one neuron away from the cochlea. The cochlea is here. All hell breaks loose. 8 different cell types, 5 different parallel processing streams, all preserving tonotopy. That is the representation of the auditory signal. And that's just the start. How do we ever hear anything? Yeah, I mean, it's just mind-boggling, the complexity that is in our nervous systems. Now, there are some things we can do. Many of you may work on fMRI, but I just want to remind you that fMRI is based on knowing whether something is happening. It's about blood flow. So all we know is something is happening. There's increased blood flow in one of these VOX cells. And each VOX cell of the human brain, so the smallest image we've got, the finest resolution we have, contains 5.5 million neurons, up to 55 billion synapses, and these figures are true, 22 kilometers of dendrites, and 220 kilometers of axons. And it's just, how we can tell anything from an fMRI scan. I mean, there's a lot of argument about that, which I'm not going to go into, but this just tells you how coarse it is. If we think about the problems of understanding that single neuron, that single stretch receptor in the maggot, this is the horror in a human brain. And above all, where is not how. It's not, I didn't say that. Famous neuroscientist said that just because you know where is something is happening, doesn't mean to say you know actually what's going on in this tiny little part of the brain, which has 220 kilometers of axons in it. Now, there have been some attempts to understand this, in particular with vision. I'm just going to give you a couple of examples. Those of you who don't know this stuff will be amazed. Those of you who do will go, yeah, well, we know that. Hubel and Wiesel, back in the 1950s, imagined that maybe we could detect lines. And we could detect lines if we had a detector that was linked to the retina that could detect a dot. This is one's here. This cell here is detecting this dot. The cell next door can detect a dot very, very close. And these cells will detect dots down there. And if you can wire these cells up together in a kind of hierarchical process, a bit like Smee's diagram, then you've got a line detector. Okay, that's fine. Now, when I was an undergraduate, we all laughed at the story of, well, in that case, if you can have a thing to detect a line, you must have a grandmother cell. You must have a cell in your brain, not only for identifying your grandmother, but your grandmother standing up, your grandmother sitting down, your grandmother riding backwards on a horse playing the banjo. And of course, that can't be how the nervous system works because the brain isn't big enough. But about 10 years ago, people discovered cells that apparently responded solely to pictures of Jennifer Aniston or the Sydney Opera House. It did look as though there was something as precise as a single cell that would only fire. These were patients who very generously gave their time. They were being operated on for epilepsy. And before the operation, they allowed researchers to go digging around and show them pictures and to record from single cells in their brain. And they found single cells that only respond, in some people, that only responded to Jennifer Aniston, but not to Brad Pitt, who she was going out with at the time, and probably not for the woman behind. Only Jennifer Aniston, or only the Sydney Opera House, or a piece of paper with the word Sydney Opera House on it. So it seems as though there's a remarkable degree of precision in our brains. Maybe we do have grandmother cells. Well, we don't. The reality is that they got lucky. They just happened to be recording from one of the cells involved in the whole network, which could be millions. We've got 80 odd billion cells in our brain. There's plenty to go around. In the millions of cells that would be slightly enable you to recognize Jennifer Aniston, but perhaps see similarities and differences with the woman behind her. Now, many of you probably may think, well, what about AI? Can't deep learning help us? Can't that provide? Maybe it's all that's the answer. We can use AI to understand how vision works. That's what Geoffrey Hinton from Google tried to do. So about 6 years ago, they gave one of their deep learning programs loads and loads of YouTube videos to watch. Well, clearly it's not actually watching them, but they took loads and loads of stills, about 10 million, I think, stills from YouTube, converted them to zeros and ones, gave them to the device and said, what do you think of that? And this is what the device thought. I don't know if you can see. It said, well, I've evolved a cat detector. You got to remember, these programs are not given a task. They are simply told to go away and see what patterns they can see. If you can't see the ears here, the head, eyes here, ears here, eye, nose, like that. See? So this machine had entirely spontaneously, or this program, entirely spontaneously developed a cat detector just because there are a lot of cat images on YouTube. And so the thing that it saw over and over again was a cat. And so it developed this kind of platonic image of catness that it was able to use to identify cat images. The problem, however, with this kind of deep learning is, as Hinton said in an interview, he was questioned, there's an entirely separate problem. We don't understand how these things work, right? Hinton, no, we really don't know how they work. So they don't understand how their programs are able to do what they do. I mean, they wire them up in a way, again, looking a bit like Smee's diagrams, but out of the hidden layer, as they call it, rather coyly, emerges the magic that is this kind of thing. Now, there are limits to this. Janelle Shane, she's an AI researcher, a fantastic book called, I think it's You're a Thing and I Love You. And she has fun with her programs. And she used GPT-2, which is a neural net that can write articles. It's now been superseded by GPT-3, which is even better. And I'm sure undergraduates the world over will be using this to churn out crap. You give it a prompt and then it just writes an article. And it's not bad. But it's got incredible limits. And she showed this by getting it to write what in the UK we call love hearts. In America, they're called candy hearts. I'm sure wherever you're from, you will have similar sweets, these little sweets with little phrases like, I love you, you're mine, be mine, forever, stuff like that. And this is what it came up with. So it's got the idea. It's got to be short and snappy, but it is thick as it doesn't understand anything. It doesn't understand context. Mouthy hamster, fart booby. I'd eat one with fart booby on it. But I just think this is so good because it shows both the power of this technology, but also its extreme limits because it doesn't understand context. Just as Hinton's program has got no idea what a cat is because it's just seeing zeros and ones, Janelle Shane's computer only knows it's got to be two words, and that's all it can do. Finally, I just want to big up, some of you will know of Eve Marder's work, but for me, this represents the challenge for neuroscience. Eve Marder has spent all of her career, she's based at Brandeis, all of her career trying to understand the lobster, not the lobster's brain, but the lobster's stomach. The lobster's stomach has got 30 neurons in it. Those neurons produce a rhythm which enable the stomach to grind up the lobster's food. And in fact, these 13 neurons can switch between 2 different rhythms. She knows everything about that. She's got the connectome. She knows what genes are expressed. She's got the proteome. She knows exactly the neurotransmitters, the neurohormones that are involved, everything about it. And yet she cannot show why those 30 neurons produce only those 2 rhythms. And above all, she can't predict, using computer modeling, what will happen if she removes one of those cells or alters its activity. So in the real sense, she doesn't understand those 30 neurons. So if we don't understand the lobster's stomach, what hope is there for understanding the human brain? Well, I think, and this is where I think I would be, I mean, this is what I study, so this is kind of biased. I think the way forward is trying to understand small brains, just like we did in the molecular biology revolution. We didn't try and understand mice. We started off with viruses and prokaryotic cells. The reductionist approach has turned out to be immensely productive. So by understanding small brains, we may be able to establish principles that we can then apply to more complicated and interesting brains. So the kind of things I'm talking about are obviously Drosophila, 100,000 neurons, maggot, 10,000, 12,000 maybe, zebrafish larva, about 100,000, Platineris, fewer than 1,000 brain neurons in its larval brain. These are the kind of model systems where we have the possibility of having connectomes and functional connectomes. That is, connectomes where you can manipulate the cells. We're getting there in Drosophila, maggot. It'll be another few years before they have that established. But already there are insights coming about how the maggot knows how to roll over, how it can do that. Because if you poke it, it doesn't like it and it rolls away. And so Martha Slashik, in particular, who's now in Cambridge, has been at the forefront of a great deal of work, along with Albert Cardona, her partner. So what this is all saying is that science, culture, and technology are intertwined. They're interconnected. We don't just do science in a vacuum. We take our metaphors and our analogies in particular from technology, and they help us discover things. They help us think about the world in a different way. But, all models are wrong. And metaphors are inherently limited because the brain isn't a computer. It's like a computer. It carries out some kind of weird computations, but it isn't a computer any more than it's a telephone exchange. So a framework, there comes a point when a framework becomes a cage. People found eventually that the telephone exchange or the telegraph metaphor they couldn't think about the brain in the right way because it was too flexible. I think, and it's not just my view, there are loads of think pieces in neuroscience journals arguing this, that we're getting to that point with the computer, that the computer metaphor, which has proved so immensely powerful, is starting to hit the buffers. We have so much data and we don't have a theoretical framework that everybody agrees on that can be used to interpret those data. Now, we know by looking at the past that in the future, technological developments will alter how we can imagine what the brain does. It enables us to think of new experiments. So at this point, everybody gets very excited and they often ask me, well, what will they be? And I used to say, well, if I knew that, I'd be very rich. But I got fed up of giving that answer. And so I've devised a futurometer. If you peer into your computer and stare very hard, you'll be able to see the future technology. Right, let's turn it on. God damn it. Come on. No. It's not working today. I'm afraid it doesn't work today. You're going to have to work this out for yourselves. And that's it. This is the book. This is the UK version. Order it from your local bookshop if you can. Otherwise, big companies are available that will take your money off you. Thanks a lot. How do I stop sharing? Do I do that?
I'll do it for you.
You do it for me. There you go.
Fantastic. Thank you very, very much for this treat of a presentation, and not only entertaining, but also very informative and exciting. We have time for a few questions. Yeah, so if you're willing to take them. Also to the audience out there, those of you who are not familiar with the format here, you have a little tab there on the bottom, ask a question, so you can ask it in there. I see
And
one
then big
here.
up the Principles of Neural Design book, which I talk about at the very end, by Simon. My name's gone a blank. His name's gone a blank. Anyway, it's a really good book. It's very interesting.
Okay.
And yeah.
Uh,
Okay. Right. Off you go.
One question here from Hernan Lopez here. I wonder whether you think that organisms like Hydra, with such simple and stereotypic nervous systems, would be the answer to the question of how the nervous system works.
Yeah, I mean, I think that could be part of the problem. Is you've got in terms of, let's say you thought that was the idea. I don't think that would be a good idea. If you put in a grant to, let's say the EU, let's say they decide we're not going to have big brain anymore, or blue brain, whatever it's called, and we're going to have a new organismal based project, and we're going to do it on Hydra. Immediately everybody would say, but wait a minute, we know an awful lot more about these other model systems. And I think developing a new model system, and the only possible exception of that is Platineris, which Jekyll has developed. He's now in Exeter, and that is turning into a very simple few hundred neuron brain model system. I think there'll be enormous weight about, well, we've got to grow the damn things, we've got to learn about them. We've been studying Drosophila for over 100 years. So there's an awful lot of background knowledge which you can just start on. On the other hand, they're very complicated. My guess is that really simple neurosystems like Platineris, like Hydra, are going to provide principles a bit like this, that emergent properties will come out of them that will be very helpful for then understanding more complex nervous systems. I think, I'll leave it there. So I think that would be a good move, but I think you'd probably get pushback from the neuroscience community. Say, oh God, not another one. And anyway, it's green. Why? That's a plant, isn't it?
I mean, beyond looking for simple system, what untapped potential do you see in comparative anatomy approaches, even comparing complex systems and subtracting the differences?
I think that's got to be the way to go. And I think that the fact that part of the problem is that our vision of anatomy has been, anatomical. So, I mean, I've forgotten the details. There was a paper a few weeks ago, and they came up by looking at using RNA-seq and looking at the expression patterns in brain cells in the mouse. I think they came up with about 800 different kinds of cell. They're able to classify the mouse brain not into the several dozen anatomically based cells, but hundreds and hundreds of genetically based cells in terms of what they're actually doing. And that, I think, is, I mean, in a way, it's even more data, so it's going to be even more difficult to understand. But that, as you say, by then doing subtractive screens, either in different species or rearing them under different conditions, that is going to provide insight, maybe into quite profound things like depression or whatever. I mean, I'm just making that up, but you can imagine rearing a mouse in very deprived conditions and comparing that, genetically identical mouse with a mouse that's been raised in rich conditions. You may well find differences that may give us some insight into changes in human behavior. I mean, I don't think that's far-fetched. If I was on a grant panel that came up to, I'd give that a big tick. I mean, it's speculative, but we need speculation. I think, there's a science that you can be confident of the outcome. It's a bit boring, but that's what we have to, that's what the panels want. They want to know what you found before you've actually found it. And I think a bit more funding of speculative that may fail, but, then we will fail better next time, as Beckett said.
Yeah, it's okay to make mistakes as long as they're new ones.
Yeah, that's very good.
There's a question about development from Sonia Garel, which relates to one I also kind of biasedly, Sonia says she's biased. Of course, I'm biased on the topic, but what about the idea that the adult brain is just the mere endpoint of a series of transient circuits that can transition, sometimes abruptly, and do end working out? Yes, so what would you say is the added value of development versus looking at the simpler stage of a complex brain as opposed to looking at a simple adult brain? Well,
given that I study the maggot, I mean, which is developing, I mean, it's not fully formed, it's turning into, it's increasing its maggotness as it gets older and is preparing to turn into a fly. So I think I think that's extremely important. Part of the problem, I guess it depends on, you want to be confident that there is a, I mean, it's going to sound terribly finalist. This is all getting very philosophical, but that, you know, there is an end point in what you're studying, and then it's not wrong, but it's not a system that's struggling to develop, that it has reached some kind of equilibrium to the extent that any system does. I think that's why I'm okay about studying maggots, because they do their thing. I think studying the developing brain in the maggot egg, in the fly egg, which people do and is fascinating, but for example, that can reveal the role of activity. You need sensory activity for the brain to form correctly, and the nervous system is learning, it's practicing as it's in the egg. I think there's a different kind of question, fundamental question, but it's telling us a different kind of thing from, say, a maggot wriggling, in the same way as watching a mouse pup walking, crawling, is telling us something different to watching an adult mouse skittling along and climbing upside down and doing all that complicated stuff. So I think development, I mean, it's something you said is virtually absent from the book. I'm afraid. I'm sorry about that. In my defense, I studied the sense of smell, and there's virtually nothing about smell either. So I was quite disciplined. I left out my area, knowing that, for example, most sensory neuroscience is visual. And I think that's a problem. I think that's biasing how we think about the brain, but that's the way it is. So I think, development. I probably should have. I mean, the book's divided into two halves, and then a brief bit about the future. So it's the past up to 1950, because I think that's when everything changes. It's when people start to use the word neuroscience in the late 1950s, and computer models become possible and start to be developed. So then there's the present, which is 1950-ish to today, and then there's the future. And the chapters in the present part are all thematic about memory or connectomes or whatever. And I guess a chapter on development would have been very interesting, but it already grew. Yeah. I ended up with 2 or 3 chapters more than I intended. And I think my publisher would have got very cross with me if I put another one in.
I think
somebody else can write that book.
I think it's worth a full book, Matthew.
Well, that's not my field, so I can't do that. I stepped outside of my comfort zone enough in this book and got very scared that I was going to get clobbered, and tried not to be too rude about people, especially about fMRI. Yes, so I allowed other people to be rude for me.
There's a question or two that relate to the scale. If I maybe summarize them, what are your thoughts, or what have been people's thought on how size relates to complexity and computational power? I mean, you gave the example of the universe-sized computer, but beyond
that was what Paul von Neumann. But you've got to remember, he didn't have transistors; he had valves. So I think the heat produced by the thing would have exploded. In fact, Smee thought the same thing. He thought that he could build a mechanical device. I didn't show any slides. He actually had. He said he claimed to have created mechanical components that could represent concepts. And he said, if I built something that could represent language, it would be the size of London, and the machine would destroy itself as soon as it started working, because it was mechanical. So, I mean, this is a kind of problem, isn't it? Because when you talk to people, and indeed students, and you say there's no tendency to increase complexity in the animal kingdom or in life, that's not the way it works. People get very cross and they say, but look, here's us. We've got these ideas in our heads, and, maggots don't. Well, I hope they don't. So there is an increased complexity, but I don't think it's linear. And if we forget about consciousness for a moment, and that's the thing I'm least interested in the book, because I think it's unknowable at the moment. I think it is knowable in principle, but I don't think any of the people trying to understand it have got there. Then clearly there is something happens. The more complex you get, then and it's not just an exponential growth, but I guess it's a kind of an extra dimensional growth in ability, in what you can manipulate, in Craig's terms, in what you can represent of the outside world. I mean, I don't think a maggot is representing much about the outside world except taste, smell, and potential movement around it that could be a threat, gravity. So a handful of sensory bits of information, whereas a fly, which has got, you know, one order of magnitude more neurons, then it's doing other things. It can start to think about other individuals and perhaps act together. And there's all sorts of interesting stuff about Drosophila about their perhaps social behavior. And certainly other insects, clearly, you know, ants and so on, have social behaviors emerging out of their tiny little brains. I mean, Darwin said that the brain of the ant was the most marvelous atom of matter in the universe, even more so than the brain of the human, he thought, because it was so small and yet could do all these astonishing things. Having worked on ants, I think I can see where he's coming from. So I think something is happening, that increased complexity and all those neurotransmitters and the wiring, because there's lots and lots of synapses at different points, that is producing extra dimensions of representation and of potential manipulation, and, if we could understand it, computation.
Mm-hmm.
But it's very striking. I asked people on Twitter at some point. Twitter was immensely useful for me in doing this. I said, what examples are there of computation in nervous systems that we really, really understand? And there were only a handful that people came back with. Everybody's confident the brain's carrying out computations. I mean, I accept that unquestioningly. But what they are in the human brain, I mean, I don't think there's any knowledge of what they might actually be. In terms of neural, you know, in terms we could actually show wiring diagram like you see in papers.
Mm-hmm.
Every paper about a nervous system has a lovely wiring diagram, which is another metaphor, of course.
Related to this, perhaps, what's your view, and what have people's view been on the most useful basic functional unit that could unite different types of brains at different times?
Well, yeah, I'm rather old-fashioned. I think I'd probably still say it's the neuron. Mm-hmm. But I mean, Eve Marder and other people, they published an article about 10 years ago in Science in which they said, we've got to go beyond that. And we've got to think about it in terms of ensembles of neurons. And maybe at a functional level, that's actually true, because I think the data on humans and Doris Tsau's work, which is absolutely extraordinary, looking at face recognition. I mean, her work just blew me away. If you don't know that, people, just go away and read some of her stuff. It is amazing on monkeys, but also on humans. And she talks about there being shifting, you know, fluctuating networks that will move in short periods of time. So you haven't got it's not like a wiring diagram at all. You know, a wiring diagram of a computer is, this is its central processing unit. This is doing some calculation, controlling the screen or whatever. But what she argues, and this feels right, is that you've got this movement. You may have a central core of neurons that are involved in recognizing a film star or whatever, but then you've got the whole pattern is shifting and changing as your brain changes, because you're thinking about other stuff, you're looking at other stuff, and information is being temporarily or permanently recorded in those patterns of activity. I mean, it all gets very hippy dippy quite quickly, I think. Mm-hmm. But I don't think there's any other option, because the wiring metaphor is so wrong, because it's fixed and rigid, and it's the telegraph. It's not the telephone exchange or the computer. You know, those wiring diagrams we see, even if they've got conditional elements in them, are still very, very rigid. And we need to think about the components changing over time, which is hard, and probably needs more complex computational models, which is, you know, outside my field.
Yeah, maybe relating to that, I was struck in the book and also in your presentation by the striking parallelism between the main technological advances of the time, so water mills, steam machines, telegraphs, computers, etc., and the way people would think about how the brains work. Do you have any vision of how people were conscious of that at the time, and whether that applies only to neuroscience, or all fields of science have also their mindset being shaped by the technologies they were using?
Well, physics is the obvious one. So physics literally had models. You know, they had orreries, those strange models which could be clockwork, in which the planets would move around, and that would predict. Newtonian mechanics which is clockwork. It's a celestial clockwork. That's what they use. The Chinese just use this to land their probe on the moon and bring it back. They didn't need any fancy quantum stuff or anything complicated. It was just Newtonian mechanics. And for that problem, that solution is adequate. On the other hand, if you want to understand anything else more complex or tiny about the physical universe, then we know that those metaphors, and just think about the billiard ball metaphor of the atom. That's what dominated, and then they realized there was stuff inside it, and so we have those things orbiting round, particles orbiting round, but they're not particles, and they're kind of clouds of potentiality or something. I don't know. So physics, I think it's very striking. Physics has given up. If you think of the words and rightly so, the words they use now are quark. They have top, up, down, but they talk about spin, which is a bad idea because it suggests that things are whizzing round, and they don't. But strange, all those words they use are deliberately chosen so they have no meaning. There's nothing else you can stick onto it. And
so that was deliberate.
Yeah. Physics and maths, they actually use a separate language, which is the language of mathematics. And neuroscience sometimes does that. I think then reconnecting it with the squishy biological reality is hard. In terms of how conscious people were in the past, I mean, Smee was very conscious. I mean, he not only thought about the brain worked that, he actually, he came up with the idea of the facts. He said, well, look, the eye is just a photoelectric cell. So if I get a photoelectric cell, and it's connected to the brain, and it's like a telegraph. So if I get a photoelectric cell that can capture an image, I can then connect that to the telegraph and send it to another city. So in the basic, using the body as a metaphor, he then developed technology. So he was well aware that the two were kind of bleeding into one another, imagining new technology on the basis of anatomy, but also using that technology to then imagine things about the brain. I mean, I don't know what's going to happen. Maybe we don't. I mean, it could all be wrong. I could be wrong. I mean, I don't think there's a rigid necessity that you have to have a machine, artificial metaphor to understand the brain. Maybe we don't. It's just that it's worked very well so far. And the impression is that we're getting, a bit stuck. I mean, I don't know how any of the listeners feel this, or people who are, I know that this isn't something I invented. I found, and in the book, there's lots of references to articles where you can see people, scratching their head, saying exactly this, we're stuck. What are we going to do next? What are we going to do with all this electrophysiological data, with the connectomic data, with the fMRI data? And it's not enough to just say, well, let's chuck it on a machine and let it chew it over, because we want to know how it works and what it's doing. And for the moment, even deep learning programs aren't going to give us that insight. So
I was going to ask you about that, because supposedly these were inspired by the structure of the visual cortex, right?
Yeah. That was the initial. There are parallels in the 1950s. There are a number of different simple computational models that are developed. So I forgot what they're all called now. They're all in my book, and they work quite well. I mean, obviously on computers that were, the size of this room and had, I don't know, not even 1K of memory. They had several bits of memory. I mean, unbelievable boldness of these people in trying to develop. Pandemonium was one of them that was developed. And their structure, which, as I say, looks a bit like the idea based on that Smee had. It's a hierarchical one, essentially, but it's this middle layer where the magic starts to work, and that's where it all gets mysterious. I mean, you may have seen that deep learning has solved protein structure, which is the most astonishing thing, that you give a sequence to this program, and apparently the program will then predict what the amino acid structure is going to be out of the amino acid sequence. What the protein structure will be. But I mean, apart from the fact that nothing's been published yet, they won't know how it works. They won't know exactly what rules this clever thing has extracted from learning about millions of protein structures and millions of genetic sequences and seeing what the link is. It can't tell us. So we can't go, oh, well, this is, we're not going to be able to work it out ourselves. So I think I think deep learning is amazing, but it's also, I think we need to be careful about how much insight it can give us into the actual processes, conscious insight that we can then apply.
Mm-hmm. It could become a model of itself, right?
Yeah, well, yeah, absolutely.
The tool becomes a model.
Yeah, I think so.
Maybe a last question here from Bassem Hassan. You mentioned, I don't know.
Hello, Bassem. Another maggot man. That's what I like. More maggot men.
You use the term. I mean, that's not Bassem's words, but at some point you used the term GUI or something, talking about all this messy stuff inside synapses. So Bassem's questions relates to the fact, or to the question as to how much of this mess is necessary for function to emerge. What's your view on stochasticity, and what have been people's view about the noise, the role of noise in generating meaningful signal?
Yeah, well, Kevin Mitchell, who I see is on here, he's been very keen on noise being a feature, not a bug. It's a key feature. And I think that's right, because, I mean, nervous systems can't do without it because they're not designed. They've evolved. And so we know that, as Francois Jacob said, that, evolution tinkers, it doesn't design, so it makes do. And as long as it's good enough to increase the survival of the, increase the fitness of the organism, then who cares if it looks messy? So we are built on such on noise and having to cope with it, and the approximate nature. I mean, I think it's down to vision. So vision looks so fantastic. I mean, it appears, except of course when you get old and then you need glasses, but it appears to be a win. Literally, you're peering out. So I think we're kind of seduced by the apparent precision of vision, whereas in fact it's not precise at all, apart from the computational business with the blind spot. It's blurry and messy, and there's virtually nothing out there. It's all here. And I think even in just idle, those of us who are neuroscientists just thinking about the nervous system forget that even sensory nervous systems are incredibly noisy. I mean, the olfactory system, which is what I work on, it still remains utterly mysterious as to what the relationship is between any particular odor and our perception. There's only a handful of examples that we've even got the slightest idea of what's going on there, including in model systems like Drosophila. So there's going to be complexity and noise, and I think they're two, to be kind of glib about it, they're two sides of the same coin. That they are both things that nervous systems both create and have to deal with in order to produce something that's good enough. And I think that's what we've got to remember. Nervous systems aren't designed. There may be some. So Simon Laughlin, that's who I forgot. Alex has put it in the chat. Simon Laughlin's book is very keen on the energetics of cells, of neurons, and how they may well be getting to an optimum. But I think that the way they're functioning overall is pretty messy, and any computer scientist who built a nervous system like one of ours would be thrown out of a job because it's so wasteful. Yeah. All this extra stuff that isn't necessary.
Why would you put the visual cortex so far from the eye in the first place?
Well, exactly. Well, and we now know, and this, so one of the things is one of the tensions, which didn't really come out too much in the book, but I think everybody will know, is the tension between localization and distribution. And yeah, things are localized, but they're also distributed. And I was amazed in rereading the Hubel and Wiesel papers, that right from the very outset in the 50s, they were noticing that cells that they were recording from that were connected in the visual cortex were also responding to sound. So there were cells that were integrating information from a completely different sensory modality. So there must be neurons going from the auditory cortex back to the visual cortex to, I mean, these were cats. So, what does a cat want to do? It wants to hear a rustle and see a movement. That's the thing it's really, really interested in. And these were cells that were doing exactly that. So there is localization of function, but there's also top-down nuancing of function. We know this in the olfactory system now as well, that the activity of the first stages in the brain are nuanced by context. There are some smells that smell, depending on, if you tell somebody, oh, you're smelling cheese, they say, oh, it's cheese. If you tell them, oh, it's something foul, it's vomit, they say, oh, it's vomit. And it's the same smell. So their perception is being altered by top-down information.
So you're saying these visual neurons responding to sound were in the very first papers?
Yeah, I can't remember which of the papers it was. Yeah, in the 1950s they noticed.
I didn't know it was that early.
Yeah. I mean, it's just kind of said in passing, and I guess like many of us, they thought this was a rather annoying artifact. Let's just put that to one side.
Interestingly, comma,
yeah. In our hands.
All right, Matthew. Thank you very, very much for this presentation, for the really lively discussion, and again, very inspiring, I think.
Okay, thank you, everybody. There've been some great questions there. I'm sorry I haven't answered them all. Yeah, I mean, I don't claim to have all the answers, and many of the insights in the book in general are not mine. They are those of the vast community of neuroscientists whose work I've shamelessly stolen with credit. I've given everybody credit. But, you know, this is my attempt from my point of view to synthesize where I think we are and to offer some ideas about where we're going, which I don't expect many people will agree with, unless they work on maggots.
Well, in any case, thanks a lot from the whole community.
Okay, thanks everybody. Stay safe. Have a great holiday. If you
Goodbye, everyone. Take care. Have a great holiday too, and see you next year. I think it's not the first, but the second week of January. I'll tweet about it. Stay safe, relax, and have fun as much as you can. See you soon. Bye, Matt. Thanks again.
Bye.